Hello, everyone. My name is Chris Mathis, and I'm the Chief Commercial Officer at AnaBios. We're going to be starting. I see people are still entering the webinar, but I will be saying a few words before I hand over the mic to our speaker. Dr. Michael Gold is a professor of neurobiology at University of Pittsburgh. Also, someone I've known for a long time. He and I were in graduate school together at UCLA in the neuroscience program a few years back. So before I get started, just want to point out a few things. If you look at your taskbar there with the go to webinar, down below where it says handouts, we do have two AnaBios brochures available. One is a AnaBios DRG brochure, and another is a CNS brochure, which has information about our human spinal cord samples and human brain samples, which is a new area for AnaBios. And if you are interested in obtaining high quality human DRG samples from AnaBios, just put your email in the chat to the organizers, and I will pass that along to someone who will contact you with information, availability, and pricing about our human DRG samples. And also, if you look in the chat, you'll see some links to our pain research library. So this is our 18th webinar in the series that we've started in 2019. So you can download previous webinars if you missed them. And there's also a link to our translational research conference, which I'll talk about in a moment. First, I just want to say a brief word about AnaBios. Many of you in the audience know about AnaBios already. But if not, I'll tell you that we are a unique contract research organization. And we're based in San Diego, California. We recover human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. We've essentially redefined first in human. So you can obtain more information from our website, anabios.com. You can download the handouts that are listed there, or you can post your email to the organizers in the chat, and we'll get back to you, especially with information about our DRG samples that are available for purchase. So it's my pleasure, before I introduce our speaker, and it really is my pleasure to moderate this session, I just want to point out the Translational Research Conference that AnaBios is putting on in February 27th and 28th in San Diego, California. Those of you, especially on the East Coast of the US or in cold climates, it's a great chance to escape winter and enjoy sunny San Diego. We've got a great list of speakers, approximately 16 speakers from different therapeutic areas. Those of you in the audience that are in pain research, we have several pain talks. Raj Khanna, who I believe is in the audience, is one of the speakers, and Ted Price is the keynote speaker. So you won't want to miss this. Registration is only $250, so you can click on the link that's in the chat and save your space now. Space is limited, so you want to make sure that you get your registration. We do also have special pricing on the hotel where this will be held. All right, so now, without further ado, I'd like to introduce our speaker. Dr. Michael Gold is a professor of neurobiology at the University of Pittsburgh. He received his BA from Berkeley, and as I mentioned, UCLA, his PhD. He was a postdoc with John Levine at UCSF. And the focus of his lab's research is the neurobiology of pain. He's made important contributions to injury-induced plasticity and nociceptive afferents, and their contribution to the manifestation of persistent pain. Toward this end, he has employed an array of approaches ranging from the study of isolated cells to the development of novel behavioral assays with which to assess the presence of persistent hypersensitivity, and more recently, the study of clinical populations suffering from persistent pain. Dr. Gold is actively involved in the greater pain community, and most recently working to form the U.S. Association for the Study of Pain. So, Michael, take it away.
Thanks, Chris. Thanks for the introduction and giving me the opportunity to speak. And hello to all you out there in the virtual audience. Hope you're all enjoying the day so far. Start out with some disclosures. We have had support from Grunenthal. I won't be talking about any of the proprietary data that we generated, but we have had grant support from them to support our, at least some of the research we've done on human DRG neurons. I want to start, because I always end up forgetting or running out of time to do so, to give the thanks to the people who actually did the work. In my lab, the folks in bold are the folks that are still there, but I'll be showing some data from people that have since moved on. But it's a great group of people in the lab, and a real joy to work with. Here at Pittsburgh, and really with now collaborators around the country, there's a number of people that have really been instrumental in giving us access to tissue, and really driving our research program forward. Of course, none of this would be possible if we didn't have support, primarily from NINDS, but we've got some money from the DoD, foundations. The University of Pittsburgh School of Medicine has been instrumental, as I noted, Grunenthal. And really, since I'm going to be talking about human tissue and data that we've collected from human tissue, it really is, one can't give enough thanks to the family members who consent to let us use their tissue from their loved ones for research purposes. And it really comes from the family members. That is, the family members have to provide consent for us to use the tissue this way. So you all ought to have these conversations at home and make sure everybody's aware of at least your wishes, and that you know what the wishes are of everybody that's part of the family. Makes for a rough transition, but we are hiring here. Want to take the opportunity to let people know. Certainly a number of my colleagues at the University of Pittsburgh Center for Pain Research are hiring, and if you hear anything interesting that's going on in our lab, we have a couple of slots open as well. And while I'm plugging things, if you all don't have too severe Zoom fatigue, and not to chase you off of this session, but starting at 12 Eastern Standard Time, or what will be 9 Pacific time, is going to be a USASP webinar with Steve Davidson and Matt Sapio, who will be following up on really the more nuts and bolts issues around this issue of procurement of tissue and how that it's used. You can register for this event through the USASP website. Raj Khanna it will be moderating. This is a basic science preclinical SIG, and he's the current chair of that SIG. So at least for the next 35, 40 minutes or so, my plan is really to address 3 major points. The first is, shouldn't be a surprise to any of you, scientists out there anyway, but there are strengths and weaknesses to every approach. I have to continually remind my colleagues who work in mice that yes, it is true of mice, and it isn't only that they are smaller than humans, but there are limitations with every approach, and it's really going to be true of human DRG neurons. Some of those limitations aren't obvious when you start out, but some really become manifest pretty quickly as you move into it. The second is that we really can and should be validating mechanisms and therapeutic targets on human tissue, and this is really the niche that AnaBios fills, is for those of you that don't have access through your institutions to this sort of tissue, that AnaBios is a tremendous source for this sort of validating and exploratory type of work. And then finally, I'll just end with a couple of slides reminding you that there's a lot more than just human DRG neurons that really can and should be used for validation and exploration. And these are complementary approaches, obviously, to a lot else that's going on that we can now do in the laboratory. So when talking about pain and analgesia, most of you are aware of this basic circuit. One can suffer a tissue injury. That's not the one I wanted, the laser pointer. One can suffer a tissue injury in the periphery. That signal is ultimately converted into an action potential that's propagated in primary afferent, or referred to as nociceptive afferents, that terminate in the spinal cord dorsal horn or the trigeminal dorsal horn. And that signal is sent up to the brain, where the perception of pain emerges. There's descending circuits that are engaged as well. So unequivocally, when one is talking about pain, pain is in the brain, and I'm not going to deny that for a second. But the underlying premise of this work is that the vast majority of clinical pain, in particular chronic pain, as will be defined in ICD-11 as pain lasting for more than 3 months, is really due to a peripheral driver. And I'm going to give you a couple of examples of this, of even the centralized or nociplastic types of pain. We have evidence to suggest that there's a peripheral driver in those types of pains as well. But if you buy this premise that the vast majority of clinical pain arises from the periphery, then looking in the periphery for mechanism of therapeutic targets is appropriate and necessary. So that sort of is the underlying premise for what I'm going to be talking about. And so obviously just about everything I'm going to be talking about is in the periphery. But as noted by Chris, AnaBios does give access to spinal cord tissue as well, which is clearly a critical first step in this sort of signaling process. I'm going to spend most of my time talking about human DRG neurons. And because I'm doing that, it's important for me to give credit where credit is due. And it was really Ina Belfer when she was here at Pittsburgh. She's now a program officer at NCCIH at the NIH, who got me looking at human DRG neurons. And she had been doing these genome-wide association studies and realized pretty early on that looking at genetic mutations and the potential causal link between sort of genes and the pain phenotype wasn't simply going to be answered through analysis of the genome, that there would be changes in the tissues that were responsible for pain that accounted for some of the variability of why one patient might have pain and not others. So certainly at a transcriptional level, which people are exploiting really beautifully now with all the RNA-seq type of data, but also that a functional analysis of these cells would be critical as well. And so we started now over 8 years ago recovering human DRG neurons to explore these sorts of issues. So what I want to address first is this issue about the strengths and weaknesses of the human DRG neuron system. And I'm going to start with weaknesses simply because many of you might be aware of the promise, but you should go in with your eyes open of sort of what you're getting into. This is a yet more simplified cartoon. This would be the DRG cell body. That they are located within the ganglia. And so when we're talking about dorsal root ganglia, this is what we're talking about here. And the peripheral terminals are out in the tissue. Central terminals terminate in the spinal cord or trigeminal dorsal horn. In humans, these can be very long neurons, right? The neurons innervating the toe of your leg can be up to 1 meter long. And so if the pain is originating in your peripheral terminals, it's got to make it all the way back. And of course, the problem here, as I've suggested, is that most of the pain is in fact originating. And when we study, that is originating in the periphery. And when we study these DRG neurons, we're studying the ganglia. And this really isn't the site. It isn't at all the site where a lot of this pain arises. That's not to say that there aren't some really interesting things going on in the ganglia. And in the last couple of years, people have really made some very interesting observations and really implicated the ganglia as a potential driver for pain syndromes. But there's a lot of evidence to suggest that that the pain for most people are experiencing is arising in the periphery. And so we're getting basically the wrong tissue for that. The second, at least from human tissue, we know which ganglia we're getting, L4, L5, L6, et cetera, et cetera. That would be lumbar levels 4, 5, or 6. And we know by dermatomes and the innervation patterns that which regions of the body are innervated by those particular ganglia, but we don't know specifically the target of innervation of any of these neurons. And in preclinical studies, you can label the tissue and you can study the properties of the tissue that's specifically labeled, or if you have the intact organ, you can look at what a visceral afferent looks like relative to a joint afferent relative to a cutaneous afferent. We and others, this is just a series of papers that we publish now, directly comparing target identified subpopulations of neurons, and the theme that has emerged through this whole series of studies is that it matters, that target of innervation matters, and that shouldn't be a surprise to anybody, given the unique functions that a neuron that innervates the skin has to play relative to one that's innervating the connective tissue, joint, or viscera, et cetera, et cetera. So we don't know the target of innervation, and that certainly is a limitation when we look at the human DRG neurons. Again, therapeutic targets, if the pain is arising in the periphery, there is the possibility that these therapeutic targets are transported out, aren't present in the cell body, and so you don't detect them in the cell body. And that's going to be true for the central terminals as well, although there is some reason to suggest that many of these proteins that would be naturally transported get inserted when you acutely dissociate these neurons. But the point is that, again, when we look at the cell body, we're looking at a site that's different from where the potential action is. And finally, well, the DRG stimulation and other lines of evidence suggest that the ganglia itself may be a reasonable therapeutic target. There's also evidence that suggests that the structure likely needs to be intact. And so we and others who are studying these neurons that are dissociated are really missing this sort of spatial and structural organization that is potentially important to how and why the ganglia may be contributing to pain in any kind of a context of injury or disease. A couple other issues, just get to highlight some of the people that collected the work here. Xulin Zhang was a postdoc in the lab who really started sort of the functional analysis of the DRG work that we were doing. Jamie Moy, it was a postdoc, just recently left, followed up on that work, and Jane Hartung followed up as well, both or all three of them doing this sort of functional analysis of DRG neuron. And it really was DRG neurons, and it was the work of these three that really has highlighted for us a number of the other issues that one should be concerned about. They weren't the first to certainly highlight this issue. We had faced this with our isolated DRG, rodent DRG neurons, but it should be clear that the isolated DRG cell body is injured. This is what the ganglia looks like when we remove it from an organ donor. The ganglia of the DRG is right here. We can get some peripheral nerve associated with it. There's some dura, some of this connective tissue, and the peripheral nerve out here. But it should be pretty obvious to everybody that we've clearly damaged the structure by removing it in this way. So we have necessarily created an injury, and when we dissociate it, shearing off these peripheral and central terminals to get these isolated cell bodies, it's pretty clear that it has been even more damaged. Now, what the impact of that is certainly a point of discussion, but it's something that people should be aware of. Human DRG, in contrast to rodent, tend to be associated with a lot more support cells, and when you acutely dissociate them, many of the cells, like this one here, are covered with these support cells that if you want to record with a patch clamp electrode, for example, you simply just can't get access to the ganglia. You sort of have to give the cells time and culture for the support cells to migrate off of the ganglia to give you clean access to the isolated cell body itself. And things change with time, as we who have studied even rodent DRG neurons know that the response to injury is not static. The other issue, particularly for people that want to record from these cells, is that the human DRG neurons are big. This is a histogram of the cell body diameter from cut sections. And versus what we recorded, just assessing the cell body from each of the neurons that we recorded. In contrast to rodents, we don't really see the skewed distribution that the rodents has, which tend to have sort of more smaller cells, and then this maybe even a second hump of larger diameter cells in the that sort of gets spread out over a larger distance. But you can see here that the average cell body diameter is something in the order of 60 microns, whereas a mouse, the median cell body diameter might be something closer to 22, 23 microns, so a third of the size. And if you look at simply the neurons that we recorded from using cell body capacitance in picofarads as a probably more accurate measure of the cell body area, it's clear that these neurons, the human neurons, are much bigger than rodent neurons, where again the median for rodent neurons is somewhere around 30 picofarads, and this would be a rat, whereas in the human it's well over 100 picofarads. So again, sort of 3 times difference in size. And the functional implications of that are clamp control. This is the response. We're looking at the fast application of GABA, in this case 300 micromolar. You get this sort of rapid inward current because we've got chloride as a depolarizing, the chloride intracellular and extracellular, such that chloride drives a depolarizing or inward current. But in this particular neuron, the clamp control is such that we see these breakthrough action potentials just on top of this GABA evoked current. And this, of course, can be a real problem if you're looking at fast activating large inward currents, such as voltage-gated sodium currents. Some other issues that come into play, we're all aware now because of, I think, the appropriate emphasis that NIH has put on this issue of sex, age, race, and ethnicity. We have some data where we've detected some mild sex differences, certainly an impact of age. We haven't seen any evidence of sort of race or ethnicity dependent differences between donors, but other factors such as the cause of death or BMI. I highlighted Steve Davidson will be talking in the next hour for the USASP session. He basically won't recover tissue from anybody who meets the sort of standard criteria for being obese, which is a BMI of over 30. This is a data set that we have recently completed, something in the order of, I guess, 78 patients or 78 donors, the distribution by sex, the distribution by age. They can be relatively young, they can be relatively old when you're getting organ donors. Cause of death can be variable, and of course BMI can also be dramatically variable. We don't restrict based on BMI, but of course history, I mean, these are humans that we're talking about, and all of us have lived pretty diverse lives with various injuries and other sorts of things that we do or shouldn't do that has impacted our overall health. Factors such as the downtime, these are people who make it into the organ donation system, and so they have been found down in one way or the other. Maybe they actually arrested or had an event within the hospital and therefore could be resuscitated relatively quickly. But in this population, 61% were found down and or required CPR, and that the mean time down was 47 minutes. So you have this variable of how long the tissue that you were looking at was hypoxic. There's the recovery and processing of the tissue can be a variable as well. In our experience, the older tissue requires much longer processing with the enzymes to dissociate the neurons, simply because of more connective tissue. The NIH released this U19 mechanisms for basically tissue banking the human tissue. Ted and his group, Ted Price and his group, and Rob Drouot and his group were recipients of these grants. And so hopefully with the data sets that they'll be generating, we'll be able to start to see the relative impact of a number of these variables on the tissue integrity, et cetera. But I bring all these issues up just to highlight, one of the last papers that we submitted to a unnamed journal was editorially rejected based on what at least by my reading is largely the assumption of the editor that that we don't know that if we see a difference between a human DRG neuron and a rodent DRG neuron, whether that's a species difference or simply because of all these other factors that are associated with processing the tissue. And so you should all be aware, humans, because for a whole variety of reasons, I mean, we've tried to do the experiments in rodents where we push the downtime out to various lengths, et cetera, that is the time at which the animal's heart has stopped beating, and what kind of recovery and what that impact has on the tissue properties. But humans, simply for a whole variety of physiological reasons, don't follow the same sorts of parameters, and so that head-to-head comparison is difficult. So that's it for the weaknesses. I'm sure there are more that I haven't touched on, but the big ones in terms of strengths, I think the strengths are obvious, but certainly important to highlight. And one of the keys is that we can study the target protein in its native environment. I just give this one example. This is an old study out of what was Abbott became AbbVie group with Mike Jarvis et al. With this NaV1.8 blocker, this A80 compound, and this is these TTX resistant NaV1.8 currents before and after the application of A80, and in heterologous expression of these different isoforms of voltage-gated sodium channels, you get this really nice separation between the selectivity of A80 for the human 1.8 relative to these other voltage-gated sodium channel isoforms, almost 3 orders of magnitude in terms of separation. The problem is that when you put, and they're just looking in this case in rodent DRG neurons, you look at the potency and efficacy of this compound in the rodent, and it isn't nearly as potent as it looks like. And when you look at efficacy, when we looked at this compound in terms of efficacy, it isn't nearly as efficacious either. So that you can do these types of pharmacological studies in the protein in its native environment, I think is essential. You can certainly confirm that the target is present and functional. This is a study out of Rob Giroux's group where they showed that the coupling, in this case metabotropic glutamate receptors to TRPV1, was different in human and mouse. But hopefully many of you have heard Rob talk about his experiences with mGluR5, where they were thinking this was going to be a great analgesic because it works wonderfully if peripherally restricted in the rodent to block a variety of pain mechanisms. But as it turns out, this mGluR5 isn't present in human DRG neurons. So the target isn't even there, which I think is just a beautiful illustration that you really got to do these studies in the human. This third strength is that you can really confirm biophysical and pharmacological properties. And I'm just going to give you a couple of examples of that. These are nicotine-evoked currents in the mouse. About 10% of mouse neurons, you see these relatively rapidly activating, rapidly inactivating currents in the rat. So it's 10% of neurons in the mouse. It's about 70% of neurons in the rat, where they're almost a 50-50 split between these fast activating, fast inactivating, and more slowly activating, non-inactivating currents in the rat. In the human, we don't see any of these fast activating, inactivating currents. We only see these slowly activating or persistent currents, and they seem to even have different pharmacological properties than in the rat. So, not just mouse and rat are different, but mouse, rat, and human are different in terms of both biophysical and pharmacological properties of nicotinic currents. Just give you another example of this. These are voltage-gated calcium currents. These are high voltage currents, and you can do an experiment where you look at the baseline current, and then you just apply these putative subtype selective blockers. Nifedipine blocks the L-type channels, conotoxin the N-type channels, agatoxin the PQ-type channels, and you can see in this particular neuron, some fraction of the currents L-type, some fraction is N-type, and another fraction is PQ type. And in the rat, there's variability, as there is in humans, in terms of the relative contribution, if you look across neurons, of each of these current subtypes. The key point of this, and whether or not it's subtle, my feeling is it isn't so subtle, is that in the rat, the dominant current is this N-type CAV 2.2 current in that dominates the high voltage gated calcium current, and in the rat L-type currents are sort of the second in line in terms of their density, and that relative percentage is inverse in the human. That is, the dominant current of these L-type and the N-type make up the minority of percentage. There's clearly variability, but on average, the bulk of the current in human is L, whereas in the rat it's N. Just an example of some unpublished data. This is NAV 1.9, or this persistent current, just a steady state and activation protocol, where these are pre-pulse amplitudes here that you can see the currents, a quick hyperpolarization, and then a step to -50. And you can see in the rat that you get complete inactivation of these currents, whereas in the human, there's some percent of current that doesn't inactivate all the way. And you can see that in these H-infinity curves, where on average, in the rat, we get complete inactivation, steady state inactivation of the currents, whereas in the human, there's about 20+% that is not subject to steady state inactivation, or at least recovers rapidly. And this, as you might imagine, should have pretty significant functional implications for a current that we view as sort of regulating the excitability based on its relative amplitude across time. We can look at second messenger properties and second messenger signaling. In this case, we're looking at the rat and the relative contribution of a current subtype. This is back to voltage-gated calcium channels, and you can apply conotoxin and then this selective mu agonist DAMGO. Conotoxin blocks a fraction of current, and when you block the N-type current in the rat, you largely eliminate the ability of DAMGO to block any additional current, suggesting that in the rat, the primary target for DAMGO is these N-type calcium channels, whereas in the human, so this would just be another example, nifedipine blocks, and again, you get a nice block still that is DAMGO blocks even more current when you've only eliminated the L-type current. In contrast, in the human, it looks like DAMGO is primarily blocking the L-type current. So not only can you look at second messenger signaling, but you do this comparison between rat and human, a lot of the way we view this GPCR signaling to voltage-gated calcium currents, this sort of fast signaling, is via this N-type channel, whereas in contrast to the rodent, and we know that because of studies of the rodent, in contrast to the rodent and the human, this inhibition appears to be primarily dominated through the inhibition of the L-type current. And it's harder to see in the way these data are plotted here, but you can largely eliminate the ability of rat DRG neurons to inhibit current if you block with conotoxin, whereas in the human, you eliminate the ability of these of DAMGO to work if you block the L-type current, and that's just the percentage of inhibition that's shown. Finally, you can do a co-expression. A lot of the people out there, as RNA-seq gets cheaper and more readily available for more and more laboratories, there's a lot of RNA-seq that's been going on. From our perspective, the question is, what does all this RNA do or mean in terms of function? And so we can do this sort of functional analysis. We can ask, how these neurons, what do they co-express functionally? In this case, we're showing that this neuron can be depolarized by applying high K. It's responsive to decreases in pH, so it potentially has some ASIC channels, although by the time you get down to 6, you might be activating TRPV1. Wasn't responsive to menthol, probably didn't have TRPM8, did have TRPA1, did have TRPV1, at least by the response to these agonists here. But comparing tissue from, in this case, a 21-month-old female, we get differences in the response to high K. We have differences in the response to acid, and we have a neuron that was responsive to menthol, AITC, and in this case, at least it may have been included, we didn't see the response to capsaicin. So not only can we look at how these things are co-expressed, but we can start to see potentially age-dependent differences as well. And what Jamie Moy did is carry this forward to look at the mRNA expression and determine the extent to which you could detect the mRNA for any of these transcripts in any of these neurons. And you really see potentially some age-dependent changes and potentially a threshold where the transcript actually means something functionally. In terms of the potential solutions to limitations, I've talked about sort of cutting off the terminals. We've been using microphysiological systems, and this is in collaboration with Hang Lin in orthopedics. His student Megan and Jamie picked this up, and Matthias has followed, where we actually can provide innervation of tissue, in this case, a knee joint tissue, the components of a knee joint grown in 3D. This is what the system looks like in reality. And we can put neurons in the middle here, human neurons that we've now cultured for weeks, and they can provide innervation of the different tissue or the joint types, so we can start to look at what's driving the activity in these different neurons visually with imaging and or on microarrays. Just to wrap up quickly with other tissue, the peripheral nerve is another tissue that one should look at. Again, Emmanuel and Jorge have been really pushing this project in the lab. Again, as I pointed out, you get the central nerve and the peripheral nerve when we take the DRG, and you can record the compound action potential in these nerves, in this case, the trigeminal and the sciatic nerve. And we can begin to look at pharmacologically, what are the ion channels that underlie propagation of action potentials in these nerves. In this case, there appears to be differences between a trigeminal nerve and a somatic nerve in terms of their sensitivity to carbamazepine. Highlight some of the work that Brad Undum has been doing in the airway, where again, from organ donors, they've been taking trachea and bronchi from organ donors and teasing nerves and recording compound action potentials. What's nice about this system is they can look at both efferent and afferent function, and again assess pharmacological properties of the afferents that are innervating these structures. People are doing the same sort of thing in the gastrointestinal tract, where you can take grind up biopsies. So this IBS, irritable bowel syndrome, is one of these disorders that's thought to be primarily driven by it's a nociplastic pain syndrome or a potentially centralized pain syndrome. But if you take tissue from these patients, grind it up, and you apply it, in this case, the DRG neurons, you get a much bigger response relative to the amount of pain that these patients have, suggesting that even in this quote-unquote centralized pain syndrome, there appears to be a peripheral driver for the pain. But even more powerful is the potential to record from some of these neurons. This is work that sort of David Bolmer and his group have pushed forward, where again, from in this case, from surgical specimens, but you can use organ donor tissue for this as well, are recording from the units from the nerves that are attached to this tissue and characterizing them based on their different properties and pharmacological properties. In terms of the bladder, this is an old study. Don't even have the pictures for the people that did this work. Xiaogang contributed to some of this. From biopsy, we can get bladder tissue from normal bladder, overactive bladder, interstitial cystitis, or painful bladder syndrome, load them with Fura, stimulate them. And what was striking is that we're putting these cells, these are just bladder epithelial cells, put them in culture, pass multiple passages over time, and we can still differentiate the cells from normal bladder and IC based on their response, in this case, to the muscarinic agonist carbachol, suggesting, at least to me, that these epigenetic changes that may be driving the pain of painful bladder syndrome are persisting. We've got some data that you can use the tooth pulp. Again, you can look at painful versus non-painful. We can see neural tissue by PGP 9.5. We can detect sodium channels, and in inflamed tissue, we get an upregulation of the inflamed tissue in the tooth pulp. People have been using the knee or the hip or the knee for the same purposes. This is just immunohistochemistry that we've collected with tyrosine hydroxylase staining or CGRP staining from tissue in the knee joint. But basically, the tissue can be used in the same sort of way. So to summarize, again, there's really strengths and weaknesses to every approach. Be aware of the weaknesses going in. It isn't the be all and the end all, but it's really powerful as a validating and potentially exploratory system. We can and should be validating mechanisms with and therapeutic targets in human tissue. And finally, there's a lot more than just DRG that can and be used, and can and should be used for validation. And I just end with a plug for the USASP annual scientific meeting. So hopefully I get to see some of you all live. It's going to be on the Duke University campus at the Washington Duke Inn. And with that, I will take questions.
Thanks, Mike. Thanks for a great webinar and presentation. Great examples of species differences. I was especially intrigued by that editorial rejection, especially in light of all of the clinical compounds that have failed because there was too much emphasis on rodent studies compared to human studies. So very interesting. So we do have time for questions from the audience. You can either write your question in the question box if you look at your panel. You can type it in there, or you can raise your hand and I will unmute you. While those questions are coming in, just a reminder that AnaBios brochures are available in the handout section, the CNS brochure, which has information about our brain samples and spinal cord samples and assay, and the DRG brochure, which has information about our samples and assays. And you might have missed it at the beginning, but I did mention that if you're interested in getting more information about acquiring samples from AnaBios, you can type your name into the chat, your email, and we will get back to you. So also, I posted links in the chat to everyone to our research library, which has access to all 18 of our webinars. And if you missed anything in this webinar, if you have to sign off early, you can go back and review it. It will be posted soon. And our translational research conference, that'll be in February, with several pain presentations. The link is there, you can register. All right, so while the questions are coming in, I'll get started, Michael. You mentioned one of the weaknesses of the DRGs is the damage to the cell bodies. Don't you think that over time, though, they recover in culture? I know that with that AnaBios, we can keep them in culture for weeks, and it seems like there would be some recovery happening there, especially with all the support cells that you mentioned.
Yeah, I do think they recover. I mean, certainly using ATF3, for example, as a marker, at least in our hands, ATF3 staining goes down within 4 to 5 days. You don't detect it anymore. But my colleague next door is Rick Kerber, who spent a lot of time characterizing the neuronal response to a crush injury, where the neurons are able to essentially reinnervate their target. And what seems to be pretty clear from that data set is that while they're regenerating, they still aren't quite normal.
Yeah.
So, I mean, I completely agree. We're using this microphysiological system with the intent on and are getting innervation of tissue. We haven't finished the characterization to see whether these neurons go from a hyperexcitable state prior to reaching their target, and then basically quiet down again once they reach the target. That would be consistent with sort of the resolution of a pain phenotype.
Right. Thank you. Okay, here's a question from Raj Khanna in the audience. He says, great overview of weakness and strengths of using human DRGs. Are there more N-type calcium channels in younger human DRGs than compared to older ones?
Yeah, so I'm trying to remember. It was the current density that really changed by age, and there was a significant correlation with the current density going down with age. But I do not recall. I'd have to look back at the paper. I apologize to remind myself on whether it was the relative proportion changed. My sense is that the relative proportion didn't change with age. Okay. So we did see a significant age effect, but I don't believe there was a proportion shift. And this is looking at, we didn't look at our very young donors simply because we don't have enough of those, but this is looking at the sort of 18 to 22-year-olds relative to a 40 plus relative to a over 70.
Got it. Thanks. Just a reminder to the audience, you can either raise your hand or type in the question box. Here's another one from the audience. Given the variables between different donors and preparation, how do you choose the number of neurons you need to patch for any given target? And do you make a distinction between voltage clamp and current clamp?
Yeah, so that's a great question. I mean, in general, my rule of thumb is I like to see at least comparable effects, comparable, whether it's current magnitudes, comparable properties in neurons from 3 donors. Sort of is my minimal threshold. That the more donors, the better. A lot of these things are sort of time limited. At least in our hands, the voltage-gated sodium current density increases relatively rapidly. So after a couple of days in culture, you just simply can't clamp the sodium currents well enough anymore to get a reasonable, at least full sort of biophysical characterization of the cells, even with very, very reduced sodium in the bath solution. So time is a bit of a factor. I mean, for a lot of the things that we've looked at, the properties, whether it be TTX resistant sodium currents, whether it be NAV, NAV 1.8, putative NAV 1.8, or putative NAV 1.9, those sorts of channels seem to be relatively consistent. Where, like I say, if we look hard at the data, we might pick up a difference in sort of density, but we don't get major differences in terms of the properties. And so, in general, 3 responses. We have had some outliers every now and then, and that's why I like to do at least 3, and then we can go back and look at the outlier. The way we get our tissue, with our organ procurement organization is CORE. It's a fantastic organization to work with, but there's a little bit of a delay between when we get the tissue and when we get the medical records. And so we have experiments planned. We'll do the experiments when the tissue is processed, and then maybe a week to 2 weeks later, we'll get the records and realize, oh, this person had, some particular issue, and that might explain, again, if we saw something different. But it really can, doing it for the very rare cases where things really look very different. If you replicate it in at least 3 donors, you can generally pick up things that are donor specific.
Got it. Thanks. Raj said, thanks for the answer on the N-type. Makes one wonder then what gabapentinoids work on. The second question I have is that you, I don't know, do you want to comment on that? Because it sounds like he has a second question.
Well, I mean, so clearly Raj is thinking that Whelan Pan may be wrong, that it's not an NMDA dependent channel. I mean, Whelan Pan's got a pretty extensive story that the gabapentin is actually acting, that the alpha-2 delta helps trafficking of NMDA receptors, and the gabapentin is probably influencing the NMDA receptors rather than CAV channel. I mean, it's the alpha-2 delta channel or subunit. If you believe that that's sort of the primary binding target, whether it's exclusively trafficking N-type or other sorts of channels, I think is open to question. And so it may be activating or acting through L-type channels as well.
Okay.
Go ahead.
I
was going to say, just as a follow-up, the sort of what's relevant, and the reviewer said, might be subtle differences. Yeah. What is very clear with sort of a rapid sort of canonical G beta gamma dependent inhibition of CAV 2.2 is it's rapid, right? I mean, it's hundreds of milliseconds. You're getting sort of full inhibition. Off rate is very fast, and that accounts for at least our understanding of sort of regulation of synaptic transmission with these presynaptic inhibitory GPCRs. The kinetics of the inhibition, DAMGO-induced inhibition of these L-type channels doesn't appear to be. I mean, we have some evidence with pre-pulse potentiation, et cetera, that there is a G beta gamma component to it, but it's much slower than the sort of canonical G beta gamma inhibition.
Got it. Thanks. So another question is, he says, I have it that, oops, I lost it, just a sec. That in addition to the variables you mentioned already, do you see changes in current and or pharmacological responses of the human DRGs in culture?
Yeah, so we've been spending, as we've been moving into these microphysiological systems, it's certainly important to us to know whether these neurons have changed their phenotype. Do we, just as a crude response, can we still detect capsaicin responsive neurons? Does the proportion change? The sort of the classic, what would be nociceptive transducers, TRPV1, TRPA1, et cetera. Do we get differences in the distribution of those neurons, particularly with evidence from rodent models that injury can result in the differential patterns of expression? And in general, our data is suggesting we're not getting dramatic changes, at least at 14, 21, 28 days in culture in those sorts of expression patterns. As I mentioned, the currents themselves, the densities can change relatively rapidly in that first couple of days. After you're sort of out a couple of days and the neurons have started to grow processes, in general, we don't try to do voltage clamp recording from those neurons simply because clamp control becomes very difficult. Current clamp recording would be a different story at that point, and or using the MEAs, in which case we can get action potentials that seem to fire just fine.
Got it. Here's another question from the audience. What is your opinion on the utility of iPSC-derived human sensory neurons as a surrogate for primary human DRGs?
Right. So at the IASP meeting, pre-meeting, I sort of gave my quick summary of a sort of the head-to-head comparison that we've been using. We haven't tried this sort of new protocol from, now I'm blocking on the name of the Atomic, that seems to be able to give you a nociceptive phenotype within a week to 2 weeks, where they get a higher density of NAV1.8, of TRPV1, et cetera. I mean, minimally, the iPSCs are smaller. I brought up this issue of size, and that size has a whole host of implications in terms of receptor coupling, calcium dynamics, et cetera, et cetera. And iPSCs, simply because they're 10 microns in size in general, they don't have the same size issues. And at least by the protocols, I mean, we've been coached by Angelica Lampert, trying to sort of follow her protocols, and we can see protein for V1, we can see protein for NAV1.8, but the current density isn't anything like we see in human neurons, and the action potentials don't look the same. Calcium dynamics look the same. So I think that we're still tweaking. The short answer is we're still tweaking the iPSC protocols, trying to come up with something that optimizes a phenotype that gives us sort of the functional proteins of interest. I don't think we're quite there yet. That doesn't mean that they aren't really useful for a host of questions. You just got to be clear on what questions you're asking with them.
Right. Yeah, that makes sense to me. I mean, our DRG cultures, I'm sure yours, there's 25, something like 25 different types of neurons in the culture with supporting cells, and so the stem cell neurons are more homogeneous. So
right.
It's just,
yeah. I mean, people have sort of criticized, oh, what do you do with all that heterogeneity? In my mind, the heterogeneity is a feature, not a bug. And in our physiological sort of, innervating the different components of the knee joint, we can see neurons that respond differently to the fat pad versus the connective tissue.
Yeah. Another question from the audience, a comment and a question. Thank you for a great and very insightful talk. Considering that it is not possible to get tissue without some damage, do you have any suggestion on how to reduce the potential influence that it will have on functional studies? How do you foresee us mimicking an intact nerve and DRG structure? It seems like you kind of alluded to that in the microphysiological system. Any other comments on that?
Yeah, I mean, this is sort of why I think one has to be thinking about their system, their questions, and not using any single approach to answer a problem, because there are strengths and weaknesses to all approaches. I think the microphysiological systems are one way to get at I mean, I know there are people that are using those systems to see whether the sensory neurons can innervate what would look like dorsal horn neurons. We're using it to innervate peripheral tissues. There are protocols now for adding back, you know, the Schwann cells, so you can actually start to get myelination and the formation of nodes of Ranvier in these processes. So you can do a lot in these microphysiological systems to recapitulate what looked like an intact system. But ultimately, you're still, it's microphysiological, right? You know, we've got, you know, our chambers are probably 2 centimeters in diameter, which is relatively big for some of these systems. And it just isn't going to recapitulate a 1 meter, you know, peripheral process. So, you know, using this in combination with microneurography in humans, where you can actually record from the nerves and stimulate the terminals that are intact, are the, you know, what you need to start thinking about in terms of how to use approaches in combination.
Yeah, you know, at IASP, a hot topic was the connection of the DRG cells to skin, or keratinocytes and so forth. There were some reports on that, Diana Minicello's group on these microchip systems. What do you think of that setup?
Oh, I mean, I think it's great. I completely agree. And, you know, I mean, Steve McMahon it's a paper I continue to cite showing that, you know, if you do a cross anastomosis, you see the properties of neurons that are innervating muscle versus skin change depending on their target of innervation. You know, I am a firm believer, and per the whole series of studies that I highlighted that we've done comparing, you know, cutaneous versus visceral, I think target of innervation is key. I think the growing body of evidence to suggest that the peripheral tissue contributes to the transduction process is going to be important, and that you can begin to tease out, you know, not only how does the neuron interact with the peripheral tissue, with that interaction changing features of both cell types, and you can do that in the microphysiological system, but you can start to tease apart how that signaling works together. So I think the microphysiological systems are great to address those kind of questions.
Another question from the audience. Based on your work, does GABA A activation modulate human DRG soma excitability?
Yes.
Okay, that was quick and easy. Very good.
Yeah, no, I mean, it causes depolarization. So I didn't include that in my list of papers, but we have a paper where we measured the equilibrium potential for chloride in human DRG neurons. It's depolarizing as in rodents, and it should function in a very similar way in terms of driving a depolarization that will inactivate, depending on the properties, you know, the other channels that are present, depolarization induce inactivation of the TTX sensitive channels, might make the cell fire more if it has more TTX resistant channels, et cetera, et cetera.
Yeah, excellent. All right, well, thanks again, Michael. Thanks everyone in the audience for attending. I'd like to also thank my colleague, Gary Watkins, our Director of Marketing, for putting this together. Mike, thanks for your commitment to the pain community. You know, this is important research that everyone is involved in here, especially in light of the opioid crisis that's been ongoing for several years. So keep up the good work, and look for us again at our next pain webinar. And it looks like there's time for people to jump onto the USASP webinar. So thanks again, and we'll see you next time. Thanks, Mike.
Thank you, everybody. Thanks, Chris.